So just so you know,
Your program is just that it's so verified.
it's Python, Perl, R, C, made up.
There is no Perl in that.
You sure? I think she evaluated.
The other stuff might be true, though.
It's so convoluted, it looks like
Hey, you think that code? Okay, I'm not going to. No. Don't go there.
I just remembered the function she has where she opens a file and writes a header.
You know, it'll be interesting to see how we all manage with mission time. I think it's going to be a bigger adjustment than people realize.
She actually has a function in her program that all
Glad
it
they're
does
doing it, but I agree
is
it's
open
going
a
to be an
file,
adjustment.
writes one
It's
line,
going to be, Mars.
that's it.
She's going to move to the end of the hour.
Closes the file.
It closes.
Yeah, right.
That's
I
all
think
it
people
does.
are just going to forget for a while. They're going to show up at 10 after.
Well, I don't think people are going to, and they're really, I actually agree with
It's
you,
called
because
like simple
if you
fun.
need the extra time to get to your next class, you can always step out of the class a few minutes early while the rest of the class can sit there. Versus if I start 10 minutes late, everybody misses that 10 minutes. So I think overall it'll extend our ability to teach while still giving students the flexibility. Unless you have that professor that puts the quiz in the last 5 minutes of class, and that would just suck. But I guess you could do that now and have the quiz in the first 5 minutes of class. So.
I think that's
All right, it's go time. All right, welcome everyone
I agree.
to
I
the
think we should
Technology
schedule
Seminar
for students
Series.
to have time
We have a
with
sign-in
you
sheet going around.
I don't
Please
think the schedule
do
is
sign in. As always, it helps us with the pizza. So I'm pleased to present today's speaker. We have Fan Zhang, who is a student in DCMB and is in Human Conflict.
Okay. Hello, everybody. Today I'm going to present one of our recent work, VerifyBamID 2.0, Ancestry-agnostic detection and estimation of sample contamination from DNA sequence data. So, should I stand here? Or?
Wherever's comfortable.
Okay. So many of you must have heard of this tool, many of you haven't. So it is a software that was designed to detect and estimate DNA sample contamination from sequence data. So far, we have already received a lot of usage report, and according to our phone home status, it has been ran more than 2.6 million times across 25,000 different IP addresses. And
That's fantastic, man.
our wiki page is also accessed more than 27,000 times. And this tool has been adopted to be a part of many standard sequence QC pipeline, and it has been using right now in like TopMap project and CCDG, and it also was used by many major sequencing center, including Broad, Sanger, and so on. So let's first explain what the problem exactly is. So when sequencing DNA sample, normally we will see, we will categorize into these 4 different configuration based on if we are studying human DNA and if it is non-human DNA, and considering the sample you are of interest and the sample and the DNA content that is trying to contaminate in your sample, you will have these different configurations. And normally we will focus on human DNA study related part, and and then later you can basically divide this into 2 categories. One is cross species and the other is within species. And most of the time, the cross species will be easier to detect because you can search your sequence, try to match it with candidate database, and you will find out those alien DNA sequences. And one of the common scenario is this, like in 2011, there were several reports that many non-human DNA genome database was found to be contaminated by human DNA. And of course, this should not be a single case, and it's not surprising that there is also excessive contamination happened within human DNA genomes. So today we will only focus on this harder problem, that to detect the contamination level between human DNAs. And one of the possible explanation is that the introduced human DNA contamination could be introduced by human activity, because all those experiments was carried out by human, and those contamination could be like pre-existed in your specimen, or could be introduced while peptiding, and also could happen in shipment, like too little dry ice, and your sample is melted, and also it could be introduced when you do the fragment size selection, and also sometimes you will come across unexpected new or weird protocol that will give you unexpected adapter, and even could be this could be happen that sometimes you will have incorrect barcode for your when you try to sequence multiple samples in a single run, and also it could happen after all these things you manage to survive, but at the end of the day you may experience some problem in silico demultiplexing, like alignment or using rounded, all kinds of stuff. Those problems are all problems we met in real practice, and why we need to those problems introduced in this complicated procedure explains a little bit why we need to pay attention to this problem. And sequencing is a much more complicated procedure comparing with array-based genotyping, and so this method is specifically designed for the sequence data, and also judging from the usage report we have received, people really care about this problem, and not only people don't brag about it because it will generate a negative result, and if you haven't heard of it. And also, most of the time, sequencing project will tend to be more, will cost more money and time. And if you can detect this problem before you're doing downstream analysis, it will save you a lot of money and time. So let's look at this problem in a ideal theoretical way. So suppose now we are looking at Zhang's DNA, and each row here is a read from his genome, and the first row is a reference genome. And this is a snapshot after alignment, and you can see the highlighted column is a place where we will call the genotype to be AA, and it is different. It is different from the reference genome, and everything is fine. And sometimes you will encounter some heterozygous site, and you will see most of the times it will be like a half-half because of the diploidity nature of our human genome. And it is also fine that if two genomes are sequenced and you didn't mix them up, and you can tell those genotypes very easily. And here, for example, we got GG and CC for Jane's DNA, and the TT and the AC for John's DNA. But however, if contamination happens, then the genotype cannot be confidently called. And here, you will get two heterozygous, and the allele, the ratio here is not even close to 50%. So to model this problem, here is the assumptions we need to do. First, our goal is to estimate the contamination level. We say alpha here, and we assume a DNA sample that may be contaminated by at most one other DNA sample, and we assume population allele frequency of common SNPs are known, and we assume the independence across different reads and different sites. And the approach we take is to use mixture model to calculate the probability of the sequence reads and allowing for the contamination. Specifically, the notation here we define is this. So denote B as the base at the specific site and the specific read, and denote E to be the arrow, genotyping arrow, or sequencing arrow indicator. And G1 is the true genotype for the intended sample, and G2 is the true genotype for the contaminating sample, and alpha to be a contamination proportion. So the probability of observing a certain base, given these settings, can be modeled as a linear combination of the two probability. That is observing certain base given each underlying true genotype. So the likelihood function for this problem can be formulated this way. So this is the probability of certain base pair we just saw previously, and then we will marginalize out all the possible sequencing error state. And then because we assume the independence across all the rays at a certain site, we multiply the probability for all the bases in a certain site. And then further, because we are considering two genomes now, we will summing over all the unknown possible potential true genotypes. And the prior probability of each of these two genotypes can be modeled as the binomial, can be modeled as the modeled by the allele frequency, because the genotype follows a binomial distribution with the allele frequency as the parameter. And if we assume it's Hardy-Weinberg equilibrium, and P here is the Fred scale base quality, which we can get from the sequencing data. And then consider we have M different sites across the genome. Then because we assume it's independent, so we can multiply them together. Then by maximize this likelihood function, ideally we will get a estimation of the contamination alpha.
Question. So you just said that you can, for that multiplication, because you're assuming that they're independent across multiple sites, but if there's contamination, wouldn't you expect perhaps individual haplotypes to not be independent because the variation would be shared on those haplotypes, and the contamination would thus be shared, so they would be correlated with each other.
So in real practice, we pre-selected those markers. So basically, we have so first of all, like we select the markers from the HapMap and tend to restrict the adjacent distance, and also like we mask out the certain regions and different certain allele frequency and certain like variant type, like adjacent indel or something. So basically, we are choosing a subset of those all possible variants.
So you're basically already subsetting by haplotype, and then you're treating the haplotypes as independent.
Yes.
Okay, I got you. That makes sense.
And this is the illustration of the previous way in VerifyBamID 1. So originally it kind of give you the option to incorporate with the array genotype, because we usually treat the original type as much more confident in the information. So we treat it as a true genotype. And now here we get rid of this information and just to use the sequence reads and the allele frequency. So to evaluate the performance, we constructed some in silico contamination. And here we selected individuals from European population and mixing them up with proportion ranging from 1% to 20%. And in total, we got 90 artificially contaminated samples with an average sequencing depth around 4x. And here is the figure that the x-axis is the intended alpha, and the y-axis is the estimated alpha. You can see most of those estimated is centered around the diagonal, which means the accuracy is good. Well, that is all. That is the work we have done on VerifyBamID 1, and but there, as we just saw, there are some strong assumptions. One of them is that we assume that we know the correct allele prior allele frequency, but this is not always true. Like for example, when you collect a sample and its self-identified ethnicity is not correct, or when the ethnicity the sample claimed is not in your database. For example, in Canada, we don't have a representative population information so far, I guess. And or even this sample is mixed. It's not that as clear as we saw in like Southern Genome. So we will need to take care of this problem. So here is a figure that's showing if we misspecify the allele frequency, what will happen. Again, the x-axis is the intended contamination level, and the y-axis is the ratio between estimated and the intended contamination level. So the red dot line will be the optimal solution, and anywhere away from it is a deviation. So the red bar is for African allele frequency. Green is for East Asian allele frequency, and the blue is for European allele frequency, and the purple is the pooled allele frequency. Like we don't, when we calculate allele frequency, we don't care about where those samples come from. And here are two sample sets. One is the finish, which we treated as a European sample set, and all of them are contamination in silico contaminated with another sample from the same population. And same goes for same is for the YRI, which is an African sample. So we can see that when we specify the correct allele frequency, like a European here and the African here, the estimation will be closer to the ideal solution comparing to specifying the wrong allele frequency. So the problem is now that the problem is in the allele frequency estimation, now we will take a step back to look at how we exactly calculated our allele frequency. So first of all, in a homogeneous population, we usually calculate our allele frequency this way. So G is the genotype. Normally we will take the value 0, 1, 2 and divide it by the total number of chromosomes in this population. And sometimes when your host sample is discretely structured, by which I mean, like in Southern Genome, again, you will have a clear classification with African people, European people, and Asian people. Now, in that case, you can calculate allele frequency by each group. And in general, that you can represent your allele frequency with weighted summation across all the individuals' genotype. So for the homogeneous population, the weight is actually just 1/2n. And for the discretely structured population, it is just an indicator function and divided by the total number of chromosomes in that specific population. How about, but this representation doesn't include like continuously structured population. But this representation, continuous representation of each sample is not new. We have already seen it like in the PC components based method already, like here. And in 2016, there are several papers introduced the idea of a individual specific allele frequency. So basically, the idea is that they use PC they model the allele frequency as a function of the PCs that you will assign to this specific sample. So what we do now is that we take the reference panel, for example, the Human Genome Diversity Panel or Southern Genome Project, and we pre-calculate the PCA or SVD of this reference panel. Then we can use the pre-calculated the projection matrix to model our specific sample that you are interested in. So here is the detailed formula. So here, the G is the genotype matrix of the reference panel, and we centered it. And then we can do SVD analysis on it. And then we take the first K principal components. Now the first K will be approximate to the original genotype matrix. And then we can use this approximation to calculate our genotype allele frequency. So yeah. So here, this G is the estimated genotype, and this is the one you observed, you sampled from it, which means that this could be a continuous value, but this one is always 0, 1, 2. And then now look at our method we talked about before. Ideally, if there is no contamination, when incorporated with the PCs, we will only estimate the PC as the parameter in this likelihood function. That is, so ideally, it will generate a PC coordinates that will coincide with other relatedness estimate method. So we compared it with trace. Trace is a method that we use often to estimate the PC coordinates of each individual. So on the left is for the trace, and on the right is for the VerifyBamID. Those points are those gray points are HTTP points, but those colored points are 1000G points. So you can see, generally speaking, those two figures are almost identical, which means that our PC coordinates estimation is accurate. And this is for the inside samples. Those are American African samples. So you can see previously here, blue is mostly a European sample, and yellow is African sample. So American African spread across this line, which I think can represent its genome content. So now that we show that the PC coordinates as I would say, a bridge between the individual allele frequency and our likelihood model is a successful case. Now we will try to make it harder to introduce the contamination level into this likelihood model. So then we continue to construct 20 artificial samples, but this time those samples are not mixed with individual from its own population. Rather, it was mixed by individual from African samples. So you can see we have 10 CHS samples mixed with 10 African samples and 10 CU samples mixed up by 10 VRI samples. And the mixing level is still ranging from 1% to 20%, and with the African be the minor portion. So now here is a PC estimation, a PC coordinate estimation in this new setting. Like now our samples are contaminated, and you can see those samples are drifting from its supposed to be originate towards the African origin point. And moreover, if you look at those color with more heavily contaminated, for example, the purple color here, it will be dragged much more towards the African points. So it means the contamination indeed affect our PC coordinate estimation. And now this brings up the question: which comes first? The PC convoluted with PC will be essential for your contamination estimation, and also your contamination will do something bad to your PC coordinate estimation. So the only way is to incorporate these two parameters in the same likelihood framework. So this is the formula above is the one without contamination. So here the probability of observing certain bases does not depending on the alpha, but here it depends on the alpha. And we also think that now that we are considering the PC origination, we would say we would consider if the two genomes that if they are coming from the same background, and possibly it will do some damage to our estimation. So here, in the within ancestry model, we assume those two samples are coming from the same background. But for the between ancestry model, we assume those two are coming from a different background. And now in total, we will have like 1, 2, 3 parameters need to be estimated together. So this figure shows that if we consider the contamination level into our likelihood model, it will correct the PC coordinate estimation. So these 3 panels are, the first one is the within ancestry model. The second one is within ancestry model, but no, the first one is the one when you specify the allele frequency. The second one is the when you assume there is no contamination. The second one is that you use a within ancestry model, but you assume that there is contamination. Then the third one is you use between ancestry model and assume there is contamination. And still, those are the sample we used previously that are contaminated by the African sample. We can see that the driftness now are gradually corrected if our models are becoming more complicated. And it is what we expected. And now that PC estimation are corrected, we would think that we would ask if our contamination level estimation is also corrected. So we constructed those samples further. For within ancestry contamination, we randomly choose 10 pairs of individuals in each of these 4 populations, and we contaminated with each other within each population respectively. And also, when considering between ancestry contamination, the intended sample was contaminated from samples from another population. For example, the configurations here we are using is CHS with VRI or GBR with Mexico, and so on. The mixing proportion still is ranging from 1% to 20%. In total, we collected 400 artificially contaminated samples, still with sequencing depths of around 4x. So now, these three panels demonstrate the result for the within ancestry contamination. Still, apart from the allele frequency, pre-specified allele frequency, here we also introduced two additional bars, which are the within ancestry model and the between ancestry model. So notice that all the data sets here are actually within ancestry mix contamination. So as we expected, the within ancestry model and the between ancestry model basically have the similar performance here. However, if those data sets are between ancestry contamination, for example, here CHS is contaminated by VRI, Mexico is contaminated by CHS, VRI is contaminated by GBR, then the between ancestry model will always be better than the within ancestry model, since we introduced a more degree of freedom in it. And basically, these two result proves our method and shows that it can simultaneously correct the PC coordinate estimation and also the contamination level estimation. And here are some computational notes. We maximize those likelihood function using Nelder-Mead method, and the between ancestry model, because of its complexity, like in default we calculated up to 4 PC components for each individual. In that case, we will have like 9 parameters in the model, so sometimes it's hard to converge. So here we first use the within ancestry model to optimize it, which will give a decent start point, and then we use those estimation as start point for between ancestry model. And in this way, we have a very nice convergence rate generally. And the current implementation consumes around 500 megabits, regardless we are using 10K or 100K variants. And it took 16 minutes and 120 minutes for each individual. And it supports both SPAN format and the CRAM format. And so in summary, our sequence contamination method is a simple mixer model-based method to allow us to detect and estimate contamination. And the accurate contamination estimate enabled more accurate genotyping for deep genome data. And the joint estimation with genetic ancestry increases robustness against population heterogeneity. And the method also works well for detecting RNA-seq or epigenetic examples contamination. I would like to thank my advisor, Hamin Kang, and Matthew, and Gu, and Gonzalo, and Mike. Thanks.
I have another question. So this is great. This is actually really cool. One of the, I don't have to say that. Obviously, all the users using it agree with me. One potential, I don't know, maybe you can tell me because I don't really know, but it seems intuitive that based off the types of contamination that you described, that collecting family samples might be, where you're collecting trios or pedigree, that might be a case where you're accidentally mixing samples or contaminating from other family members. I imagine those would be a lot harder to piece apart due to the relatedness. Have you looked at that at all? And how well would you imagine this would work?
We have to look at within family contamination because that's too close.
It seems like
You
that
see that
would be.
within ancestry is
The
already
Ancestry seems like, yeah, but within Ancestry seems like that would be difficult enough. But
Yeah. But however, we have another modification or extension to this model. We will call it VerifyOmics. It's in developing, and actually it will now. So far, I've got the data, and it can most of the time successfully distinguish between the trios or parents or cousins and basically those closely related relationship. Yeah. So based on that, we can further distinguish.
cool, thank you.
So it seems a strong assumption here is that you'll only get contamination from one person.
Yes.
Like, do you have like any expectation that that assumption can be relaxed?
So I think to extend that to multiple sample is straightforward, but there are two problems. One is that you will introduce way much more parameters. It's hard to convert. Second is that when there are multiple samples, the effective information for the third or maybe the second sample will not be sufficient to detect. So basically, it's hard to distinguish if the contaminating part is from one sample or two samples.
I guess the thing is that for that kind of cases, we don't really care. Or like,
Yeah, you
for
can say it that
certain
way.
studies, we don't really care about like who are the contaminators, right? So
Yeah.
The most important thing I want to know is like how much is being contaminated,
Yes.
like that ratio. And I don't even care about the ratio for individual contaminators, or I only care about the ratio of the main sample that or the major components of my sample. Like,
Yes.
do you see a way to extend that?
So, in my opinion, as long as you don't care the rest of part, like the contaminating part, the contributing part, that carefully, it might not necessarily improve the accuracy of your contamination level estimation. Like, for example, if you have 4x and only one base pair you observed, and you assume there are two samples, how can you distinguish them apart, right?
So since this is a mixture model, I'm guessing there's some DAG underlying it, direct to acyclic graph
Yes.
Bayesian model. So is the only parameter you're inferring alpha fraction? That's the only unknown parameter?
The BC coordinates.
And the PC coordinates, okay.
Yeah.
How are you inferring the parameters? Are you using expectation maximization or what?
We tried, but it's hard to derive the closed form. So we just use the other mid.
Okay, and how are you assessing accuracy besides simulation?
Currently, we don't know that except for
That's always one of the
artificial
trickier.
intelligence.
That's always one of the trickier parts with unsupervised learning models. You
Yeah.
you just say accuracy, but you're really only assessing it with data, simulated data, where you already know the answer.
Yes.
So then you have a known knowns, unknown unknowns
But,
problem.
Yeah, of course, that's a general question. But in our simulation procedure, we didn't have like really that much relaxed or strict constraint on it. Right? It's a natural mixing thing.
Can you think of a natural way to design an experiment, not a simulation, but a wet lab biological experiment, where you can already know the answer ahead of time and assess your models?
So
Just think of a hypothetical experiment.
actually, our data is already very high level. It's not from like a marker wise or base pair wise. It's just basically we mix up two true data set.
Mm-hmm.
So that is so far that is the way most obvious way to do it, I think.
So I'm just asking, is there a way you can do that instead of simulating on a computer, do that in a wet, do the same thing in a wet lab experiment?
Oh.
Because there's always a difference between theory and practice. Of
Right.
course, in theory is the same as practice, but
Well, when you want to introduce contamination in wet lab, if you want to measure it before you mix it up, then your sample is consumed, right?
Mhm.
And when you mix it up, how can you, like, you don't have a wet lab way to evaluate this beforehand, I assume, right?
You can titer it, right? Like, in fact, we actually, I think Jeff Kidd's group, we did this looking as a way to measure somatic mutations, right, using known genotypes. And we took some different cell lines and just titered them together at certain percentages. We could probably share that. I don't know if we did a WGS or just the make pairs on that, but if we have it, might be a good way to look at it in a physically mixed sample as opposed to a synthetically mixed sample.
Okay. That's where we need expert in wet lab.
Well, I am not that, but I could point.
The other thing is that, following Daniel's question, we apply within ancestry model and between ancestry model onto real data. Do you see a huge difference between the inference results?
Yeah, there is.
No, the one that you show is from simulated data. I'm talking about like real sequencing data. Because that would be the case.
There is.
And that's going to ask is like for most commonly protocol.
So yeah, we use this method on top map dataset, and the behavior is similar to this figure on real data. So basically, within ancestry, normally we'll try to inflate a little bit.
Is there any requirement for sequencing depth?
We only tried 4x. We don't know the bottom line where we can go. Okay, then thank you, everybody.